In this section, we're going to look at the control of plasma lipids. And the first thing we should do probably is talk about what the normal metabolism of lipids in the blood looks like and why we should want to control it. Now, let's just get something out of the way here. When we're talking about lipid metabolism, we're going to be talking quite a lot about two main types of lipids. We're going to be talking about triglycerides, and that's a triglyceride molecule model that you can see there. And we're also going to be talking about cholesterol, which is quite a well-known name nowadays. So the triglyceride, also known as triacylglycerol, consists of a backbone of the molecule glycerol, which you can see down the left-hand side here, attached to 3 fatty acids. And during metabolism, this molecule can be cleaved in order to release those 3 fatty acids from the triglyceride molecule, and those fatty acids can then be used by cells to provide energy. And in fact, they're a richer source of energy than glucose is. They yield more energy per molecule. So let's start at the beginning and talk about how lipids actually get into our bloodstream in the first place. You can see here in this diagram that we've got a small intestine up here with lots of globules of fat, which are in fact small emulsified droplets of fat because they have been combined with bile salts, which you can see shown here as little purple things, which have emulsified the larger globs of fat into much smaller droplets of fat, which can then be more easily worked on by the lipases, the fat digesting enzymes that are released into the small intestine. And so in the small intestine, the emulsified fat droplets get worked on by lipases, and the lipases get hold of the triglyceride molecules and split them up by enzymatic action into fatty acids, as we've just seen, but also monoglycerides, his glycerol with just one fatty acid. So smaller lipid molecules that can then be absorbed by the cells of the small intestine. What happens then in those small intestinal cells is those fatty acids and monoglycerides get put back together again into triglycerides. Who'd have thought it, eh? So they get reassembled into larger molecules, the triglycerides again, and then they get packaged up in big bundles of triglycerides and cholesterol, covered with a coat of phospholipids and proteins. So they get bundled up into these things that are known as chylomicrons. And that is the form in which they are absorbed, not into the bloodstream, but into lacteals, in other words, into lymphatic ducts in the form of these chylomicrons. Now, the reason for this, of course, is that fats are not soluble in watery substances, which, of course, the blood is, and therefore they have to be transported through the circulatory system using some kind of transport system because they won't be soluble and therefore they can't float around on their own. So this creation of chylomicrons in the cells of the small intestine then leads to their absorption into these lacteals, which take the chylomicrons around and eventually empty into the venous circulation. So they do eventually get into the blood in this form. And here's what the chylomicron looks like here. It's enormous compared to some other lipoproteins that we're going to look at now, but you can see it looks a bit like a sphere made of cell membranes, and a lot of what you're seeing here are phospholipid molecules interspersed with free cholesterol molecules, but inside is packed with triglycerides and cholesterol. And this thing that's looped around the outside of the chylomicron here is actually a protein. And these larger molecules here that you're seeing studding the outside of the chylomicron are also proteins. The chylomicron, as it turns out, is not the only way that we transport our fats around. Chylomicron starts off the process, but then you get various other types of lipoprotein formed, and they are in descending size order. Very low density lipoprotein here, intermediate density lipoprotein here, then low density lipoprotein, and finally high density lipoprotein. And the chances are you will have heard about these two lipoproteins, the HDL and the LDL, because there's been a lot of talk about them. But what you can see when you compare all of these ways of bundling up fats is that they have a very similar structure. They're all spherical, but they're just getting smaller. And you'll see in a minute how what's inside changes as they go through these various stages. But you can see from the outside that in terms of the proportions of protein to the entire volume of the sphere, it's much greater in these smaller lipoproteins. So that's what we're referring to when we talk about the density. With a high density lipoprotein versus a low or very low density lipoprotein, we're talking about the density of the proteins on the lipoprotein. So you can see that here the density of the proteins is quite low. This looks like a little bit of string just draped across. Compare it with the LDL, where that protein is now taking up quite a lot of the space. It's much more dense in terms of protein. Okay, so all of these are ways of transporting triglycerides and cholesterol around the circulation, and they perform different roles. And as you will probably have heard, LDL and HDL are talked about the most, and usually LDL is referred to as bad cholesterol and often HDL is referred to as good cholesterol. Those are rather inaccurate names, as you'll find out, but they are indicating that LDL tends to have a damaging effect on the blood vessels, and it contributes to the development of atherosclerotic plaques, whereas HDL definitely has a protective effect as regards the health of the blood vessels. And of course, that's why we're talking about the control of blood lipids, because it's about the control of lipids that are damaging to the blood vessel and why they're damaging and what we can do about it. So here's a lipoprotein with a little slice taken out of it so we can look inside and see what's packed inside. Here's the outer phospholipids, the odd cholesterol molecule dotted around, and here's these big chunky proteins that are wrapped around. So this is looking like an LDL, a low density lipoprotein. The proteins are actually known as apolipoproteins. You'll see that quite a lot, apolipoproteins draped around the lipoproteins there. But in this section that's been cut out, what you can see is the inner core, which is packed with cholesterol molecules, which are these yellow ones, and also triglycerides. So a mixture of triglycerides and cholesterol is packed into the core of your lipoproteins. Different proportions depending on which lipoprotein you're looking at, as we'll see in a minute. Different lipoproteins, and as it turns out, different tendencies to damage the blood vessel and therefore to lead to or to accelerate the development of atherosclerotic plaques in blood vessels. And whether a lipoprotein does that or not will determine whether it's called atherogenic or not atherogenic. Atherogenic means it contributes to the development of atheromas or atherosclerotic plaques. So let's just have a look at the relationship between these different lipoproteins and why and how they're damaging. Starting with the chylomicron, this is, as we've seen, produced in the cells that line the intestine. It's the form in which your fats are absorbed eventually into the circulation via the lacteals. It's very large, as you've seen, and therefore it's not atherogenic because it's too big to burrow through the endothelial layer of the blood vessel and start to cause problems, because what we know is that to get an atherosclerotic plaque, you have to get the fats going under the endothelium, perhaps at a point at which the endothelium is damaged slightly, and the chylomicrons are too big to do that. So the chylomicrons are not atherogenic. So the chylomicron circulates around, and as it goes around, an enzyme called lipoprotein lipase starts to break the triglycerides in the core of the chylomicron down and cleaves off the free fatty acids and releases those into the circulation, and they can be used by cells. So the whole point of the chylomicron is to take the fats around in a transport form in order that when they get to the cells, an enzyme can free up the free fatty acids and they can actually be used for energy. So what happens, of course, when the lipoprotein lipase gets going on the inner core is that you gradually deplete triglycerides because you're gradually taking the free fatty acids out. Here are some triglycerides still intact, and here the free fatty acids have been broken off. And what happens is the core becomes more cholesterol than it is triglyceride. The triglycerides are blue and the cholesterol molecules red. As you start to break down the triglyceride and release the free fatty acids, you get less and less triglyceride in the core and relatively more cholesterol, simply because that's not being released. And the result of that is that the chylomicron gets smaller and shriveled up, and it becomes known as a chylomicron fragment. And what is mainly left in the core after the triglycerides have been broken down is cholesterol. Now, at that point, that fragment has become small enough to cause a problem in the blood vessel. In other words, cause atheroma to develop. So that chylomicron fragment that is left is considered atherogenic. Now, what happens to that chylomicron fragment is it is actually taken in to the liver, and then they are broken down, and then the liver can use the components of that fragment to make other lipoproteins and do various things with it. And once you've taken it out of circulation, then it isn't atherogenic anymore, obviously. But here's something important to notice, and that is that in order for any of these lipoproteins to be translocated anywhere, they require to be able to dock onto special receptors in the liver cells before they're taken up. And so this is where these proteins that we were seeing on the outside of the lipoprotein come into play, because they fit the receptors on the liver cells and various other places in order to interact with those cells. So docking occurs between one of those proteins that's on the outer surface of the lipoprotein and a specialized receptor, in this case, on the liver cells, and that allows the liver to take these fragments out of circulation, which is a good thing.